Good afternoon, everyone. Thank you for joining us. Welcome back. I'm Steve.
And I'm Scott.
And together we are Backyard Musings, the Science and Technology channel.
Yep. And this afternoon we're talking about a health issue, which we always like to talk about, especially when it is a positive health issue. And this is.
Yes. Preliminary studies indicate that derivatives of verticillin A can kill certain types of glioma cells. MIT chemists have, for the first time, successfully created in the laboratory a fungal molecule called verticillin A. This compound was first discovered more than 50 years ago and has been recognized for its potential as an anti-cancer agent. Would you like to know more?
Absolutely. I think everybody would like to know more. Although verticillin A differs from some related molecules by only a small number of atoms, its complex structure made it much more challenging to synthesize than those similar compounds. Quote, "We have a much better appreciation for how those subtle structural changes can significantly increase the synthetic challenge," says Mohammed Movasagi, an an MIT professor of chemistry. Okay.
Keep going.
Now we have the technology where we can not only access them for the first time, more than 50 years after they were isolated, but also we can make many design variants which can enable further detailed studies. And that was also a quote.
I wonder if they did it with AI.
Maybe so.
In
That seems to be catching on.
Yep. In experiments with human cancer cells, one modified form of verticillin A showed strong activity against a rare pediatric brain tumor known as diffuse midline glioma. The researchers caution that additional testing will be necessary before its suitability for clinical use can be determined. So send me your money is what they're saying.
Yeah.
Movasagi and Jun Key, an associate professor of medicine at Dana-Farber Cancer Institute, Boston Children's Cancer and Blood Disorders Center, and Harvard Medical School, are the senior authors of the study, which was published on December 2nd in the Journal of the American Chemical Society. Walker Noss, PhD 2024, is the lead author of the paper. Zhiqi Wang, a medicinal chemist and chemical biologist at Dana-Farber, and Marielle Philbin, research director in the pediatric neurology oncology program at Dana-Farber Boston Children's Cancer and Blood Disorders Center, are also authors of the study. Woo!
They need to pay these guys more, so they only have to have one job.
Yeah.
It's crazy. All right. Researchers first reported the isolation of verticillin A from fungi, which use it for protection against pathogens in 1970. Verticillin A and related fungal compounds have drawn interest for the potential anti-cancer and antimicrobial activity, but their complexity has made them difficult to synthesize.
In 2009, Movasagi's lab reported the synthesis of plus or minus 11,11-dioxyverticillin A, a fungal compound similar to verticillin A. That molecule has 10 rings and 8 stereogenic centers, or carbon atoms, that have 4 different chemical groups attached to them. These groups have to be attached in a way that ensures they have the correct orientation, of course, or stereochemistry, with respect to the rest of the molecule. That's elementary, my dear Watson.
Stereochemistry, does that mean you're hearing it from both sides?
Yeah, both sides.
Interesting. One synthesis was achieved. However, synthesis of verticillin A remained challenging, even though the only difference between verticillin A and plus or minus 11, what is that, 11 feet? Can't be feet, right?
No, symbol.
Deoxyverticillin A is the presence of 2 oxygen atoms.
Oh, that makes sense.
I mean, I learned that in 3rd grade.
I know.
I
know. Quote, those 2 oxygens greatly limit the window of opportunity that you have in terms of doing chemical transformations, Movasagi says. It makes the compound so much more fragile, so much more sensitive, so that even though we have had years of methodological advances, the compound continued to pose a challenge for us, and the compound could have its feelings hurt too. So sensitive.
Both of the verticillin A compounds consist of two identical fragments that must be joined together to form a molecule called a dimer. To create the plus or minus 11 feet, whatever that is, I don't even know what I'm reading here. Didioxyverticillin A, the researchers had performed the dimerization reaction near the end of the synthesis, then added 4 critical carbon sulfur bonds. I could just imagine a scientist who knows this stuff listening to me and saying, man, if you only knew what the heck you were talking about. But we're not the messengers.
So let's
we
just
are the messengers. Sorry, we
let's
don't
just
know.
call it Diddy verticillin A. How about
Okay.
that?
Diddy, there you go.
Yet, when trying to synthesize verticillin A, the researcher found that waiting to add those carbon sulfur bonds at the end did not result in the correct stereochemistry. As a result, the researchers had to rethink their approach and ended up creating a very different synthetic sequence.
They had to rebalance the stereo. They had to go from the left side to the right side to balance that a little
Took them
bit.
50 years, yeah.
Yeah. Okay. Quote: what we learned was the timing of the events is absolutely critical. We had to significantly change the order of the bond-forming events. This is according to Mavasaki. The verticillin A synthesis begins with an amino acid derivative known as beta hydroxytryptophan. Tryptophan? Okay. And then step by step, the researchers added a variety of chemical functional groups, including alcohols, ketones, amides, and in a way that ensures the correct stereochemistry.
That makes so much sense. I mean, now it makes so much sense. It's too bad it took 50 years to find this out. A functional group containing 2 carbon sulfur bonds and a disulfide bond were introduced early on to help control the stereochemistry of the molecule. But the sensitive disulfides had their feelings hurt, had to be masked and protected as a pair of sulfides to prevent them from breakdown under subsequent chemical reactions. See, even little atoms like this, they break down and they cry. You know what I'm saying? The disulfide containing groups were then regenerated at the dimerization reaction. Of course.
Okay, quote again. This particular dimerization really stands out in terms of the complexity of the substrates that we're bringing together, which have such a dense array of functional groups and stereochemistry, Mobasagi says. The overall synthesis requires 16 steps from the beta hydroxytryptophan starting material to verticillin A.
Now, if you've made it this far, stick with us for another 5 to 10 minutes, okay? Stick with us. Once the researchers had successfully completed the synthesis, they were also able to tweak it to generate derivates of verticillin A. Researchers at Dana-Farber then tested these compounds against several types of diffuse midline glioma, a rare brain tumor that has few treatment options.
The researchers found that the DMG cell lines most susceptible to those compounds were those that have high levels of protein called EZHIP. This protein, which plays a role in the methylation, okay, of DNA, has been previously identified as a potential drug target for DMG.
Cool. Quote, identifying the potential targets of these compounds will play a critical role in further understanding their mechanism of action, and more importantly, will help optimize the compounds from the Mobasagi lab to be more target specific for novel therapy development, Key says.
The verticillin derivatives appear to interact with EZHIP or EZHIP in a way that increases DNA methylation, which induces the cancer cells to underprogram cell death.
Oh, yeah.
So killing the cells, right? Killing the cancer.
Yeah.
The compounds that were most successful at killing these cells were sulfolate nilated. Again, back to the plus minus 11 feet. What did we say that was going to be?
Diddy verticillin.
Verticillin A, and N sulfonylated verticillin A. Geez. N
Sulfonylation. You there? Oh, Scott's not there. N-sulfonylation, the addition of a functional group containing sulfur and oxygen, makes the molecules more stable. Quote, the natural product itself is not the most potent, but it's the natural product synthesis that brought us to a point where we can make these derivatives and study them, Movasagi says. The Dana-Farber team is now working on further validating the mechanism of action of the verticillin derivatives, and they also hope to begin testing the compounds in animal models of pediatric brain cancers. That's awesome. Quote, natural compounds have been valuable resources for drug discovery, and we will fully evaluate the therapeutic potential of these molecules by integrating our expertise in chemistry, of course, chemical biology, cancer biology, and patient care. We have also profiled our lead molecules in more than 800 cancer cell lines, and we will be able to understand their functions more broadly in other cancers, Key says. So there you have it. I'm glad you lasted this long. Scott, something happened to Scott. Hopefully a radiation bomb didn't go off. We'll figure that out. We'll see you in the morning. Go Army, beat Navy.